like to eat a fish lot, but I don't know the battle between the fish species until I'm into this project. As you say, all the fish species in their local habitat live happily, extend their generations, and does not have any hindrance. But when the monster comes, it stop it generation, it consumes a lot. So one of the monster is like northern pike. It is invasive species in the region of southeastern parts of Norway. It consumes a lot brown trouts and mussels, and it puts the species into the endangered category. So our concern is we should identify this at early stage. How we can do that? By doing eDNA monitoring. So the DNA studied by the living organism into the environment in form of tissue, blood, or eggs, we collect that as a DNA and process it for identifying the target. So the traditional method of doing eDNA monitoring is collecting waters from rivers or lakes or any other source of water bodies. So we have to collect liters and liters of water and bring it to the laboratory. And it also has certain criteria to be followed on the day of collection, like pH and temperature, salinity, everything should be monitored at the time of collections. So after collection, we go for filtration. The filtration is for making the DNA to settle on the filter bed. So we took the filter bed for processing the next step of extracting DNA from the filters. So as you see, it's a kind of lot of mess to be handled. It is also more stressful. So to avoid this mess and stressful, we brought the filtering unit into the on-site and did our filtration directly from the lake. So it, for some, to certain extent, it was like less stressful for us. Only we filter the DNA and bring it to the laboratory and do the extraction process. So one of the most used method of extraction is chemical method. We follow the same. Here we agglomerate all the proteins and everything, and the debris, everything is removed by repeated washes. Finally, we get the pure concentration of DNA. So this everything should be handled by an expertise, not with a common man or anything like a unknown person. It also has a lot of waiting time. It is happening in a single day. So it wasn't the aquatic ecosystem because already the invasive species makes its population. So it is making worse on the ecosystem. So the researchers come up with an idea of automatic monitoring ecosystem. My supervisors had a vital role in designing the automatic ecosystem, and I'm delightful in the part of it. We designed a prototype called a Fortetech machine with all things in the to make it functionable. If you see this one, it looks like a single instrument, but it has a multi-discipline on it to make it functionable. We have electronics on board, the fluorescent overhead, and the outer system, and everything. Finally, this is the heart of the system. We call it a Saligo cassette. So the cassette is covered by top and bottom layer. Inside, the chip is made of a lab-on-chip theme. So once the sample is loaded on the one chamber, it flows inside the chip by automatic scripts and does all the extraction process and detection of the target species. So if you see, this is the extended functioning of the chip. Like if you load the sample in valve 4, it automatically goes to the valve 2 for the washing, one by one. And finally, it will go to the top layer. That's a reaction chamber. We can identify the target, what we want. Our concern is pike, so we found pike in our study, like whether it is positive or negative. So it happens in an hour. We no need to wait for like a day or something, so we can take immediate action after our results. So we performed the study in Bore Lake, which is located in Vestfold. We collected samples and processed it, and finally, we found that in our samples, we found the target pike, E, but in our sample, we couldn't see salmon. So our study representing that lake has a pike and a eel, but there's no salmon on it. So another concrete problem is we can't store the reagents which to be used for the amplification in the room temperature because it easily degrades. So our answering for that storage of reagent in the room temperature is like making all the reagents cofactor ions in a frozen bead, like a lifeless bead. So we synthesize this bead with all the enzyme primers we designed for pike and the cofactor ions and everything with a protective agent called triglyceride. So we did freeze drying and stored it in room temperature for kind of time, 1 month, and we studied for stability. So over a period of time, when the beads are stored at room temperature, it is active. But when stored at 4 degree, it was active for 1 year. So we found the difference between like when stored at 4 degree, it was more active than when stored at room temperature. So we recommend this bead to be stored at room temperature without any interventions for automatic sampling techniques. So this is the result we got from a 4-detect system. We are happy to see this result because we used our bead to detect the target species from the water we collected from the Borel lake. So the fluorescent signal which goes above the threshold, so we consider this as a positive. When it stays below, then it is a totally negative. So it happens in an hour. So as in a group, we had some few steps to succeed to make our prototype end product. So we are on the way. Yeah. Thank you all for the listening.